increase in the diameter of the capillaries. First thing that happens is you have vasodilation because you have injury. The next step is that you are going to increase the permeability in the capillaries, allowing the escape of plasma and plasma proteins. Your third step are friendly little leukocytes. What are leukocytes again? White blood cells. Thank you for answering. From the capillaries and their accumulation of the injury at the site. So they're going to come in and clean up the debris. Okay? So inflammation continues. Okay? We have localized infection, spread of microbial invaders, neutralization of any toxins that are produced at the site, and we're going to try to repair the damage. The symptoms, as we just stated, redness, heat, swelling, and pain. 4 symptoms of inflammation. Okay? That does not mean infection. It simply means inflammation. Inflammation occurs if this process does not continue, or if we have not, our second line of defense has not been able to manage those invaders, those pathogens. Okay? And you have a pretty little picture in your book about inflammation. Okay? And that's what we tend to see. This is the process and the sequence of events we just finished going through. Okay, we have tissue injury that leads to vasodilation, increased cellular permeability, the emigration of the leukocytes or white cells. Okay, we have this chemotaxis that's sending out those cytokinases, our cells that send out those chemical mediators, cytokinases that call all the phagocytes on board. Let's go, let's go. And then the phagocytes arrive and munch up like little Pac-Man. Eat up all the bacteria, pathogens, viruses, or anything else that has entered the body that they can handle. Okay? Chemical debris sometimes actually goes to the next level and produces all sorts of lovely exudate. What is exudate? What's exudate? Drainage. Usually we're talking pus. Okay? Now, is pus necessarily bad?
No.
Why not? Because most of us get grossed out by pus. So why is pus not always bad? What's in pus?
It's the cells,
White blood cells.
bacteria.
Yeah. So what's in pus?
White blood cells.
Lots of white blood cells. Hence why they're kind of got this greenish white coating to it. Lots of white blood cells. So seeing pus is not necessarily the worst thing in the world, because what it's telling us when we see pus is the body has truly mounted that second line of defense. The cytokines have done their job. They've called out the leukocytes and they've said, come and to this area, we need every one of you guys to come down here and try to take care of the bacteria or pathogens, whatever's in there. So pus in and of itself is not the worst thing in the world. Okay, it just means we've had an overwhelming response, and that's a good thing, not a bad thing. Okay, so exudate is usually thick, greenish yellow, containing many live and dead leukocytes. Once we have lots of it, we call it purulent. Just a lovely word for a whole lot of it. Okay, so the inflammatory response has kicked in. In chronic inflammation, obviously we don't see that. Okay, we have a different pathway that we take, but in chronic inflammation, like in arthritis, you're not going to have purulent exudate. Pus-producing microorganisms are called pyogenics, and strep and staph are big known pathogens that cause pus to grow. Yum. Put that on a slice of bread. Okay. So what is phagocytosis? What is it?
When phagocytes eat up the
Yeah, phagocytes. And what are phagocytes? Should be, let's see, page 260, table 15.1. They're white cells, yeah. Should be table 15.1 on page 260. That's what I have written here. You have a whole bunch of stuff on there. Yeah, there we go. Steps in phagocytosis. Okay, that's where we're heading. Okay. So phagocytes are white blood cells, leukocytes, and they're found both as monocytes or lymphocytes, and they're part of the granulocytes. So we have different types of granulocytes. Those are our eosinophils, our neutrophils, and our basophils. What's the number one white blood cell? What's the most prolific? The one we, the neutrophils. Yeah, absolutely. Neutrophils are your number one. They're the ones that are going to come out in the acute inflammation and infection. Okay? Basophils and eosinophils are more the ones that are going to come out in your chronic inflammation. And the most important one of our phagocytes are our macrophages. So mostly macrophages and neutrophils are what you're going to see. Okay? And they have different cellular elements. Excuse me, in the human blood. Okay. This is what they kind of look like. They're represented what's in human blood. We have our red cell, which looks like a donut. Our let's see, where's our granules? These are our granulocytes, and you can see they have different looks as they go along. You have your basophil, your neutrophil, your monocyte, your eosinophil, the lymphocytes, which are part of the lymphatic system. And then, last but never least, is the platelet. And they are all involved in the cellular response. Okay. Here's your phagocytes. So this is the process, the steps, the 4 steps in phagocytosis. First, you have chemotaxis. And that's when the phagocytes are being attracted. We have chemotaxis, the cytokines have kicked in. Cytokines have sent out, cytokines are neurochemical mediators, have sent out the alarm. They've rung the bell on the firehouse. Okay, and they've told all the phagocytes, come on over here. And that's chemotaxis. It attracts the phagocytes to the area. And the phagocyte then attaches itself to the object of its desire. In other words, those pathogens that have now attacked and have been tagged, the phagocyte will attach to it. The next step is that it's going to, through the method of pseudopedia, which just kind of works like an amoeba, kind of crawls along. So what I always think about when I look at this is Pac-Man. Okay, it's literally going to envelop the pathogen, surround it, and take it into itself. So it literally sucks it in. And through the different organelles that the white cells have, they're going to have chemotoxic agents in them. They will literally dissolve that pathogen through enzymatic action and through chemical action. Okay, and then it will disappear. And this is kind of, see, I'm telling you, it looks like a Pac-Man. Here's our bacterium that has been identified. Okay, and this is called opsonization, this process of phagocytosis. So the bacterium has been identified, the phagocyte comes along. The bacterium has been tagged with some sort of receptor cells. Okay, the pathogen, the phagocyte's going to come along, it's literally going to attach to it and suck it in, like this. It literally will envelop it through this whole method called pseudopedia. It literally envelops it within the body, okay? And then through it becomes a phagosome, and it will continue to ingest it. And through different chemicals, the lysosomes inside the phagocyte will break down the bacteria so that it's digested by the phagocyte, which at that point is called a phagolysosome. I'm never going to quiz you on these words. Okay? And that's how phagocytosis works. Now, what happens if that doesn't work? Because it doesn't always work, right? We all know that. So what happens if that second line of defense has sent out all its armies and it didn't quite work?
The immune system will try to...
We will eventually get to our third line of defense. Now this, it's somewhere in your book, it tells about ways that the pathogen can escape, okay? So one of them is that it has this capsule. If the pathogen has a capsule that is anti-phagocytic, the phagocyte may ingest it, but the capsule protects it. The phagocyte's still going to sit there and the pathogen's not, it's going to eventually get spit back out before the blood cell will actually die, but the pathogen stays intact because the capsule keeps it living, especially those that are spored. Okay, those that have spore capsules, very hard. The phagocyte cannot get through it. And the phagocyte has a lifespan. So once the phagocyte dies, that capsule has protected the pathogen. The pathogen still continues to be able to replicate. Okay? Some bacteria, pardon me?
So then it becomes symptomatic in the body again. So it could lay like dormant.
Either dormant, remember we talked about that prodromal period of that, that time between infection, between ingestion and infection. That'll come to me the word I'm reaching and I can't think of it. I apologize, my brain truly is having problems this morning. I woke up with the wickedest headache and it's still there. Hence my second pot of coffee going for third. I just have a really wicked headache and I'm trying to teach, so I just apologize if I'm a little offbeat, or more offbeat than usual. Okay. It'll come to me in a few minutes. Some bacteria produce exoenzymes called leukocytins, which actually kill the phagocyte. So what's going to happen is that phagocyte is going to go through the same process, and that phagocyte is going to ingest that bacteria, but that bacteria or pathogen is going to say, no, boo-boo. You can't get me. I'm going to get you, and produce an exoenzyme and kill the phagocyte. And now that pathogen is going to be able to replicate because the phagocytes can't do anything. Some bacteria are not destroyed within the phagocytes, such as tuberculosis. Because the tuberculosis is so hard to kill by our natural immune system because it doesn't encapsulate. Okay? And let's see, other pathogens use other pathways to get in. Okay? And again, we're not fully understand how they circumvent the system, but they do. So we've gone through the first line of defense, we've gone through the second line of defense, we're not up to the backup team, which is our third line of defense, okay? The effects of these pathogens that are going to have once they've gone through first and second line of defense is leukopenia, okay? So we've gone through the second line of defense. Our phagocytes have come out, our white count has gone up, we've done everything. Now it gets to the point where the body cannot produce enough white cells, enough phagocytes to keep up with the demand, so we're not going to drop our white count. Okay? So in inflammation and infection, what we would see first is a rise in the white count that says, oh yeah, the body's mounting a response to infection. And then if we don't surmount the infection and get over it, what we'll see is a nasty drop. So having a very low white count is just as bad as having a really high white count, because it's an indicator that the body has mounted a response that has not come back from it, and that there's still some sort of infectious process going on, that the body cannot produce enough white cells to attack the invaders. Okay? That's the biggest one. Okay. So these are some of the effects that can impair host defense mechanisms. Okay. So being malnourished, being underweight, and even being severely overweight taxes the immune system. Increasing the levels of iron. Okay. By maintaining a normal or higher iron count, okay, we are actually protecting ourselves from infection. Okay. Stress. We all know that during times of stress, we're going to be more open to infection because it affects our immune system. Cancer and chemotherapy. Okay, the body's already being taxed by the cancer. Chemotherapy very often will decrease our white count, which makes us immunosuppressed. Genetic factors, age, the very young and the very old, okay, are certainly going to have more problems with infection. Okay. We certainly know our patients with AIDS, they've already gone through HIV, which means that they're infected by the virus. By the time you get to AIDS, you are actually fully fulminant, and AIDS, HIV and AIDS, we certainly know affects our immune system. That's what it attacks. And definitely drugs. Patients that are on steroids, glucocorticoids, patients that are on steroids are going to be immunosuppressed because it affects the immune system. So these are all different defense mechanisms that are going to be affected by, that are going to affect the immune system. So now we're going through stage 1 and stage 2. First line of defense, second line of defense. So we're going to go to the third, which is chapter 16. For those of you that are trying to keep up. Chapter 16 is covering, as you can see, it takes 2 pages to do an outline. Chapter 16 covers the third line of defense, which is what?
The immune system.
The immune system. So understanding how our immune system works will help us to understand how we protect ourselves from infection. So now, just to start from the beginning, what is the purpose of the immune system?
To protect
How does it do that? How does it protect us?
when a virus enters the body, it's fighting it. So
Yes, absolutely. Fights against infection. But how does it know? How does the immune system know which are invaders and which are us? Which cells are invaders and which ones are us? How does it know? Is there somebody there telling me?
Self and non-self.
It identifies self and non-self cells. That is the principle of the immune system. Okay? The immune system first must recognize self cells, and we each have self cells, meaning all of the cells that make us who we are, make us our bodies, are self cells. They're all individual and special to us. Okay, they are our personal fingerprint. So our immune system has to be able to identify our self cells to then know which cells are not self cells or non-self cells. And that's what our immune system does. It recognizes non-self cells and says, oh, wait a minute, that doesn't belong here and we need to deal with it because they're not invited to this party called the human body. Okay? These cells, immune system cells, originate in the bone marrow. Okay, they're lymphocytes derived from bone cells, from the bone marrow, and they're B cells, T cells, and natural killer or NK cells. Okay, these are the 3 different types of immune cells that our body produces in the bone marrow. They are the leukocytes. The lymphocytes. I said leukocytes and I meant lymphocytes.